Good morning, and thank you for joining us. I'm Laurie DaJose. I'm a science writer with Caltech's Office of Strategic Communications and an alumna of the Institute. It's my pleasure to welcome you to Conversations on COVID-19. In addition to our Caltech community, I'm pleased to welcome the Pasadena Public Library members who have joined us today. This series is hosted by the Caltech Science Exchange and designed to give all of us a chance to hear directly from the researchers in our community who are bringing insights and solutions to the COVID-19 pandemic. I am joined today by postdoctoral scholar Dr. Christopher Barnes. Christopher's research centers on antibodies, how they physically interact with viruses, and which shapes of antibodies neutralize viruses most effectively. While at Caltech, he has worked in the laboratory of Pamela Bjorkman, and he will be starting his own laboratory at Stanford in the fall as an assistant professor. Christopher, thanks for joining us.
Thank you for having me.
Yeah, so let's start off with the basics. What is an antibody?
So antibodies are nothing more than proteins that our body produces. These are produced by our immune system in response to specific antigens or pathogens, whether this be a virus, bacteria, or even pollen. And they typically have this Y-shaped form, in which the ends of the Y are used to grab on to that pathogen. And then once attached, it can either block the pathogen or the virus, in this case, from infecting your cell, or it can signal other cells in the immune system to respond and to dispose of that antigen. Typically, or I should say antibodies come in 5 different flavors or different types: IgM, IgA, IgG, D, and E. And so they all have very different functions and come out or produce at different time points throughout our adaptive immune response to an infection. But for the most part, when you hear about antibodies in the media or within your doctor's office, they're typically referring to antibodies of the isotype known as IgG. And so these are antibodies that typically are produced within 7 to 10 days upon encountering an infection or virus. And they play a very important role in our adaptive immune response in both neutralizing the virus or bacteria, as well as giving us some memory to be protected when we get challenged again by that same virus.
Yeah, so if the body has to encounter the virus first before it knows how to make the antibody, how do vaccines teach the body to make protective antibodies?
Right, so that's a good question. I mean, so I would answer it by saying, if you think about what I just told you about the Y shape, right? So you have these 2 arms of an antibody that can go out and touch, right, and grab on, hook on to the antigen. And so these arms are very specific to that type of antigen or virus being presented. So once that occurs, there's 2 main possibilities that can happen. One, the antibody, when it attaches, it basically blocks the virus from infecting the cell, or it basically just latches on and sends out a beacon to other cells in the immune system that can then now come and either engulf that virus and get rid of it, or it can detach on to the antigens presented on the cells that have been infected and are now presenting that same antigen, right, that same protein of the virus on the surface of that cell. All right, so in the same sense, the antibody can go in and attach to it and then dispose of it that way. So, in the case of when you think about a vaccine, right, and what antibodies, why do we need to how do antibodies function in the sense of a vaccine, right? So there's kind of 3 things to think about when it comes to antibodies, and that's the specificity. And so antibodies, specificity, diversity, and memory. And so antibodies are produced by cells in our immune system, and there's a diversity of these cells circulating at any given point in our body. Because of this diversity, you can get very specific immune responses when you're exposed to or challenged by a certain virus or antigen. And because of this, your body then drives that response that's very specific to that antigen and begins to make more of those cells that recognize it, right? So what we're trying to do with a vaccine when we're producing antibodies is to give a piece of a virus, right? In this case, when we talk about SARS-CoV-2, give a piece of the virus known as the spike glycoprotein. And so that's that large gray-shaped protein over my shoulder, that is on the surface of the virus. right? So we're giving our body a piece of this, and those B cells, or those cells that are very specific and can recognize that spike protein, begin to proliferate, begin to expand, and we start making more of them. And so by making more of them, now when we are actually challenged with the virus, like when we're out in our public after being vaccinated and we see the virus or the virus enters our body, our immune system has more of these cells available now to now go and attack and, and create antibodies that will be specific to encountering that spike protein. So that, that's what we're trying to do in the case of a vaccine is to expand this very specific, population of cells that give rise to antibodies that will then help protect us once we're vaccinated.
Got it. So some of us may have heard in the news about antibody cocktails. And so what are those and how do they work?
Yeah. So again, when you think about antibody cocktails, they're basically referring to these IgGs, right? These very specific antibodies. And so these IgGs can have very potent abilities at blocking or neutralizing the virus. And so, but the thing is, because they are individual proteins, there is potential for the virus to avoid or escape from that, that treatment. All right. So we all know about antibiotic resistance, and we know about, how, obviously how bacteria evolve to avoid, antibiotics. Viruses over the course of an infection have the potential to evolve, especially in chronic infections. In the case of SARS-CoV-2, probably not as much. But there is a possibility that the virus could begin to evolve to escape individual protein, right? Individual target of the antibody. So the reason there's antibody cocktails is because the more antibodies you have within this mixture, within this treatment, the harder it is for the virus to find ways to escape and to escape this this pressure being put on it by those antibodies. So an antibody cocktail is nothing more than one or two or more antibodies that have been produced in the lab with known properties and known binding. We understand the mechanism of binding and how it functions in regards of the immune system.
Got it. So you recently published research in collaboration with Rockefeller about antibodies and their effectiveness against these SARS-CoV-2 variants. So can you discuss this work and kind of the role that antibodies and variants play together?
Yeah. So our work here in Pamela's group in collaboration with Michelle Nussenswack's group at Rockefeller University, we wanted to explore really what was happening after natural infection. And from our end, we're very much interested in the mechanism of action, right? How are these antibodies that we are isolating from these recovered individuals, how are they functioning, to neutralize the virus? And what can that tell us about, potential for escape, as well as, ways we can engineer these antibodies to be a better therapeutic. So what we do here in Pamela's group is we utilize, structural biology techniques. Most prominently imaging technique called single particle cryo-EM. And in this case, we're taking that spike protein once again, and we're mixing it with these purified antibodies, right? So these are antibodies that have been isolated from people. Their sequences at least have been isolated from people, and we have now synthetically or recombinantly expressed these proteins within our own lab to understand exactly how they function. So we take these really nicely potent, these really strongly neutralizing antibodies, and we complex them with this spike protein. And once we have this complex, right, now we can look at it and image it and say exactly how is this antibody targeting the spike? Is it targeting in a way that's going to block it from interacting with its the receptor it needs for entry on our human cells, on the lung cells, or is it blocking it or is it binding a different region that may prevent it from undergoing a conformational change, right? The change is necessary for entry. So through our work, we can begin to understand exactly where these antibodies bind, how they function in neutralizing the virus. And then we can also, if we do this for enough antibodies, we can begin to tell a complete picture of what's happening within our own immune response upon infection. And so all this work, what this ends up leading to is our understanding of how to best combine these antibodies, right? Now that we know exactly how they bind and how they function, we can begin to think about ways in which we can combine them and to make these, these therapies. So for instance, the one antibody shown here over my shoulder is an antibody that targets the top of the spike protein, targeting a certain region called the receptor binding domain. And this is a part of the spike protein that's involved in entry because it's what ACE2, this receptor on our human cells, is what is necessary for it to enter into the cells. And so what this antibody does is it basically latches on at that same site, right? It overlaps with those same, the same part of the spike protein that is responsible for entry. And by doing so, it can prevent ACE2 from even binding. And on top of that, this is even an antibody that actually can lock that spike protein into this closed conformation, never even exposing, that site for entry. So through this, through our understanding of this, we can begin to say exactly why this is such a good antibody, and move these antibodies into the clinic.
Got it. So some antibodies are better at their jobs than others.
Yeah. I mean, there's a whole array of antibody potencies and neutralization, and even antibodies that we consider to be, weak, as a single antibody within the polyclonal mixture, right? Within the mixture of antibodies that you would see actually after infection, these antibodies can act, within this polyclonal mixture to be very potent and prevent, immune escape as well as neutralize the virus. And I think that's something that important to talk about because I think you mentioned the variants and how our antibodies, deal with these variants that are coming about. And so this goes back to the idea that antibodies are very specific. And so when you think about, again, the specificity of antibody, it's going to make itself, so specific to a certain protein. So that way you don't have, these reactions with other proteins that may be circulating in our body, right? So you want to be very specific, right? So in the case of these variants, what's happening is that, the virus has evolved in these sites to make basically a charge switch. It was going from a negative charge to a positive charge. And that charge switch, for one of these, variants, is a critical, route of escape, in that now the antibody that was very specific to a negative patch, all of a sudden is seeing a positive patch. And that positive patch may allow, the virus to escape because you get, the interaction won't be as strong, and the antibody won't work as well. So that's what's kind of happening with some of these variants with, again, the single antibody treatments. But when you think about the polyclonal mixture, and I think that's really important to point out, within the polyclonal mixture against these variants, we do see reductions, but you still have a very robust response because you have other antibodies targeting other parts of the spike.
Got it. So kind of coming at the virus from multiple directions.
That's right.
Okay, so with the vaccines that people are now receiving, they are presenting the body with a piece of, the genetic information for a piece of the spike protein. So are the vaccines eliciting, are the vaccines able to get your body to produce specific antibodies, or do people's antibodies kind of vary from person to person depending on their response to the spike protein fragment?
Yeah, so I mean, I think there's a great deal of diversity of a response that's going to be elicited by any one individual. No one's going to be exactly the same. But I think what is important to note about the vaccines is that everyone is producing, after 2 doses of both these mRNA vaccines, you're seeing a very strong, robust response among all individuals who have been vaccinated, right? So that tells you that we're all eliciting really good antibodies. And so recently we worked to understand exactly if the antibodies elicited by the vaccine are functionally similar to those seen in natural infection. And what we showed was, yes, they are. We see the same types of antibodies that that we were able to pull out from recovered individuals as those that were vaccinated. So this does suggest that we are making functionally similar antibodies, and those antibodies are going to act in the same manner to neutralize the virus once you are challenged or once you come into contact with that virus.
Cool. So where can people find out more about receiving these sort of antibody treatments if they're curious? Like what's the status of these antibody treatments?
Yeah, so the antibody treatments, they do exist, and I think it's important to let people know that there's a couple of ways you can define these antibodies. And so one is through a couple of hotlines. So I can give you the number. So the CVS monoclonal antibody pilot hotline is 866-316-0264. So if you call this number, you'll potentially be able to get in contact with people that can point you to where you can receive infusions for these monoclonal or these antibody cocktail treatments. In addition, Eli Lilly has a hotline, and their number is 855-545-5921. And again, this is for the Eli Lilly antibody cocktail treatment, which has been shown clinically to be effective. And so there are, these two hotlines you can call, as well as a locator tool. It's called covid.infusioncenter.org. Again, covid.infusioncenter.org. And this will help you find places where you may be able to go and find antibody treatments. And these antibody treatments are, at the beginning of this, we thought they were going to be very limited in their, because it takes a lot of effort to produce these antibodies for delivery as a therapy. But I think it's less about the production of it, but more so about the delivery of these antibodies and just getting into infusion centers and getting these into people who are at risk. All right. So if you're at risk, with comorbidities and haven't been vaccinated, and you were to contract, a SARS-CoV-2 infection, turning to these hotlines and these infusion center locators may be, useful for you to find antibody treatments, which are known to be effective. The earlier you give it, the better, the more effective they are. So I would definitely tell people to try to get these treatments if possible.
Got it. And so just to clarify, that's if you've already been infected. These aren't like preventative treatments from.
Right. So they have not been tested for prophylactic treatment, which is the preventative measures, for prevention, I should say. Obviously we have some preclinical data in animal models that suggests that they would be protective, and if given as a preventative measure, but that has not been widely clinically tested with these antibodies, but they have been clinically tested for the treatment of SARS-CoV-2 infection. And again, the earlier you give these antibody cocktails to people that have been infected, the better the outcome, the clinical outcome will be.
Great. Okay, so switching gears just a little bit, you were working on the HIV virus before the COVID-19 pandemic. So what was it like making the transition to working on the SARS-CoV-2 virus? And were there any similarities and differences in between the work?
Yeah, so, I like to tell the story when I give these seminars that I've been recently giving, sitting in group meeting maybe a year ago, actually, maybe a little bit over a year ago. And Pamela just, kind of offhandedly said that we're going to write a supplement to one of our grants to work on SARS-CoV-2. We see it's ongoing, and this pandemic that's happening in China at the moment. Just to begin to understand what's happening. And I was like, oh yeah, I'll work on that. That sounds like a fun, new, exciting venture. I didn't know it was going to end up being like this. But in hindsight, I'm glad I raised my hand to volunteer to work on these structures. And I think we were able to do this because, as structural biologists, we have worked out ways to image and understand antibody, antigen complexes using this technique, single particle cryo-EM. So the advancements in this technique allow us to quickly, shift, as long as we could understand the biochemistry, make the proteins that we wanted to study, and form stable complexes. Then the imaging portion of it with the microscopes and the facilities we have here at Caltech allowed us to transition quite easily and rapidly. And so really it was just a matter of getting the samples, getting the proteins produced, and then working out some conditions, that, as structural biologists, we have to work out in order to get, atomic structures. So yeah, it was a shift that wasn't too difficult given the similarities between, these envelope viruses. So spike, the coronavirus is very similar to HIV, and there's an envelope virus displaying a surface glycoprotein. So that is very similar, and that allowed us to quickly shift in our ability to learn more about this virus.
Yeah. So what are some big open questions in the field of structural biology right now? And what are you hoping that structural biologists and immunologists might find answers to, say in the next decade, 10 years?
Yeah, I think that's a very good question. I mean, obviously, as a structural biologist, if you look at the number of structures we have available to us, um, around all the different viruses and potential disease-causing agents, and how they interplay with our immune system and our bodies, that information is very limited, right? So crystallography has been the main driving point, another technique, but that wasn't amenable to these large protein complexes. So the onset of the revolution in the cryo-EM field has really been impactful for structural biology, because now we can begin to image these large complexes, these complex structures, without having so much protein. So really the hard part about structural biology is really the biochemistry, right? Making a stable complex, making something that is physiologically relevant. And so I guess some of the major outstanding questions in the field, you could say, is how are all these components of our immune system functioning? How not just one protein in isolation, but in the context of the cell, right? How are these proteins functioning in their native environments? And can we see different signaling events and capture them in the moment to help us understand exactly why certain cells end up having this fate while other ones have a different fate. So there are many different questions that structural biologists can address, and I hope to address moving forward.
Yeah, thanks. So a couple of weeks ago, you participated in a Pasadena community webinar about the vaccine rollout and COVID's impacts on the Black community. Can you talk about that event a little bit?
Yeah, so I was approached by the city to participate in this event. Because of there's a certain degree of hesitancy within certain communities about vaccination and treatments. And while this hesitancy is real, it doesn't completely tell the entire picture, which is something I pointed out in that panel discussion, which is we need to have more equitable practices for vaccine delivery into communities of color, and because hesitancy doesn't completely explain the low numbers of people of color being vaccinated. And so our discussion that day was really to help people understand exactly what is the vaccine, how they were tested, and letting people know that these vaccines are very safe, that they are very effective. Because unfortunately, there's a lot of misinformation circulating on social media, particularly even within the news. Articles get picked up, that and taken out of context. And so I think, as scientists, it's our job really to go out and help people understand. I mean, obviously there's a lot of information incoming for even us as scientists to deal with. So how can we expect the population and the communities to parse through this data and understand it themselves? So I value those types of events. I value this type of webinar where it allows us as scientists, those doing the work, to get in front of the community and to explain exactly what we're doing and how our work will benefit you and your families and friends. So yeah, I really appreciate the invitation from the city for that event. And I also learned a lot. I mean, I learned a lot about public health here in Pasadena, and I think they're making great strides to make sure that people are being vaccinated in an equitable fashion.
Yeah, thanks. So just one more question before we move on to the audience Q&A. You're starting your own laboratory at Stanford in the fall. Congrats. So what are your plans for your research at Stanford?
Yeah, thank you. It's an exciting time to start my own lab. And, I do plan to continue, as a structural biologist. So that's who I am. I've been trained as a graduate student and now as a postdoc. And I'm going to use my tool set and skill set actually to begin to continue to understand exactly how viruses, work to infect our cells, how we respond to those infections. And do this through a structurally driven approach. And so specific viruses, I mean, there's so many. There's a number of emerging viruses that, I think we should begin to prepare for, including other coronaviruses, work that we'll continue to do related to the SARS-like coronaviruses. But, what's not talked about is MERS. MERS is another coronavirus that is still circulating in certain populations. Obviously, there's, hemorrhagic fever viruses that, require our attention, and vector-borne viruses. And I think that is something that I'm very much interested in, because as, we deal with a changing climate and animal populations and, populations that carry these vector-borne diseases and viruses begin to shift further and further north away from their tropical climates because of the warmer climate environments that they're seeing now in the south, in the southern U.S. I think there's going to be a true reckoning with our healthcare system when we're now having to deal with uptick in cases of West Nile and these yellow fever type viruses that are vector borne. Yeah, so my research program is going to be centered around structural driven approaches to understand how viruses infect and basically the viral life cycle from infection through replication, and how we can best target specific parts of this life cycle for treatments and potentially vaccines.
Great, thank you. Okay, so we will move on to questions from the audience. So we have a question. For the students in the audience, could you talk briefly about your story? How did you get interested in science? Why biology/chemistry? And any special teachers in your life?
Yeah. So, I mean, I guess a cliche answer is always, I was always, interested in science and interested in the world around me and understanding what was going on. I do have like my microscope as a kid, my telescope. And then as I got older, I went to college with the mindset of I want to become a MD. And I think, this is something that I like to talk about, especially in communities of color. When we hear the word doctor, we rarely hear about research, doctors. We don't hear about PhDs. We think of doctor as, MD. And so that was my mindset. I mean, that's all I had ever been exposed to. I had never been exposed to research at an early age. I just had an interest in the world around me and thought that by becoming a doctor, I could begin to understand the world. But once I got to college and my grades went down from all the activities I was involved in, I turned to a professor to figure out a way to make my dreams a reality. And the answer was, well, why don't you join a research group? And that'll help people understand that you can do science, and that'll help bolster basically your applications when you apply to school. And so that led me to the lab of Gary Pilak at UNC, Chapel Hill. And, and honestly, I owe Gary a lot of who I am today is owed to him. He saw something in me that, I had yet to see in myself. And he really inspired me and pushed me to be better. Whether it be my grades, whether it be in my handling of people and things, but he really pushed me as a solid mentor, and he challenged me to think about ways in which I could impact the world. He was like, you could go to med school. He was not a huge component of going to med school, but he was like, you could go to med school, but there you may just be repeating procedures every single day. It's not a bad life. I mean, obviously, healthcare workers are very, their job is very heroic and they deserve all the credit. But he challenged me to say, you can either repeat it, or you could be the one driving new therapies, driving those treatments through research and discovery. And that, really changed my perspective on whether or not to pursue a MD or to pursue a PhD. And I like the idea of the challenge of science, of really figuring out exactly what is going to cure HIV, what is going to cure and give us a broad vaccine against influenza. And so that really is what led me to my current career path. I continued with him and then went to a PhD program, and now I'm here at Caltech, where Pamela has also been a huge driver and support of me in mentoring and helping me grow as a scientist. So surrounding yourself with good mentors, can be tricky, but it's paramount, I think, to really grow as a scientist and get you to where you want to be.
Yeah, thanks for sharing that. So we have more questions about antibodies. Someone wants to know, how long do antibodies from the treatments last, and do they last as long as the antibodies that the body produces naturally from having COVID?
Yeah, so that's a good question. It depends on how the antibodies were engineered. So we can engineer antibodies to basically have longer half-lives, which means they last longer within the body. And so in the case of HIV, we know we've done this because we would like to use some antibodies as preventative treatments, right? So you don't want people to have to come in and get an infusion every month. So we've, groups have made mutations to make the antibodies last as long as up to 3 months at a level that will be protective. I'm not aware if Eli Lilly or Regeneron made those mutations to make the antibodies longer lasting in their rollout. But in general speaking, again, these aren't considered used for preventative treatment yet. So given the doses that are administered, they would be effective at treating and neutralizing the virus during an ongoing infection. And then eventually they will wane over the course of a few weeks. So yes, I don't know if they have those mutations to make them last longer. But when it relates to natural infection, obviously you have a huge expansion of cells that's going to produce these antibodies, and in response to this infection, so you're going to have a huge influx of these antibodies circulating in your serum, and then over the course of months, these antibody levels will wane, until you maybe encounter the virus again, or boosted with a vaccine.
Yeah, so a related question is, why do some diseases result in lifetime immunity and others need to be boosted from time to time?
Yeah, that's a great question. I think Pamela got the same question, and the answer is we just don't know. Um, uh, I think there is, still a huge part of us that want to understand exactly why do you get long-lasting immunity with certain vaccines, whereas other ones you need to be boosted. Honestly, we just don't know. And so that's an open question that, I think the field will continue to research in understanding exactly why certain viruses do that and other ones don't.
Yeah. So you mentioned earlier that the immune system might react to things like pollen. So there is a question, how does the immune system distinguish between a bad actor versus a benign actor like food, and know to develop an antibody for it?
Yeah, so that's, I guess that's a great question, and it's a very involved question. And obviously, there's, antibody production and proliferation is part of our immune system, and it works in with other parts of our immune system. And so one thing about self proteins and not fight auto antibodies that would target yourself, is because we have something called tolerance that we build up over a lifetime, right? So obviously there's a number of proteins. Our proteins sometimes change, and so you don't want your body to, of course, attack itself and have these autoimmune diseases. And so there's a thing called tolerance that allows the avoidance of certain antibodies to target certain certain proteins or lipids or things, and that we come into contact on a daily basis. but then from there, when you talk about encountering antigens, so typically antigens have very distinct and very different. They look very different compared to what proteins are found in our body, and because of that, you're going to have this response, to those proteins. So, for instance, bacteria, of course, we have bacteria in our body, but when you get bacteria infections, they're going to signal a number of different immune components to attack it because of the fact that the bacterial cell wall looks very different than what we typically see in our body. So there are different ways in which our body can recognize these foreign antigens, and it does a very good job at doing that without obviously being detrimental to our own health.
Got it. Thanks for explaining that. So someone wants to know, are antibodies stored in the blood or elsewhere? And is there a limit to how many antibodies or types of antibodies that can exist within one human?
Is there a limit? I mean, we make a lot of antibody a day because it has to fill into all the spaces, right? So, when you think about IgGs, they're mostly found in our blood, circulating. And of course, we have 5 liters of blood. So, I mean, we're making a lot of antibodies, per day. But then, yes, you have other certain types of antibodies like the IgAs and IgMs that are found more in tissues in the mucosal layer, right? So the mucosal layer is a huge, surface area that antibodies have to reside in to protect us. So any given day, I don't know exactly the amount, but it was some really large amount of antibodies that we are producing and can be found in our body, to cover all these spaces between cells and within these mucosal layers to protect us from invaders. So what was the other part of the question?
Where they're stored and, oh, sorry, I lost the question. Where they're stored and if there's a limit to how many of them can exist within a person.
Okay, yeah. So yeah, I think I hit on where they're stored. So right, so they're stored and they're found really throughout multiple tissues and within the blood, within tissue, within mucosal layer in our body. And production-wise, yeah, I mean, obviously you're going to continually produce antibodies. On any given day, you can pull out antibodies. And I think throughout the course of infection, right, you're going to obviously expand those cells. They're going to be specific to that antigen. And, you can continue to produce those, antibodies, in the case of chronic infections, you continue to produce those antibodies against that antigen. And hopefully, eventually, the antibody will win and be able to neutralize the virus. But, there is no limit in which you can, continue to produce antibodies towards a very specific antigen. And I mean, I think that's kind of what HIV is, right? HIV, you have a chronic infection and left untreated, you will continue to produce, antibodies throughout the course of the infection, until the virus obviously, it attacks cells within the immune system. And so eventually, it will destroy the immune system and stop you from making new antibodies. But, throughout the course of the treatment, when you have an effective or a responsive immune system, you can continue to produce antibodies.
Got it. Another question. In order for a virus particle to be neutralized, do all of its surface spike proteins need to be bound by antibodies?
No, that's a great question. And the answer is, we don't know. And I think, that points to, outstanding questions in structural biology, right? So one question we have related to HIV and can relate this also to the spike, to the SARS-CoV viruses, but exactly how many spikes are necessary for entry? Is one spike necessary or sufficient, or do you need 3, 4, 5, 6 spikes to come together to get enough energy to then lead to fusion between the viral and host cell membranes? So first you have to understand that question before you can answer the next, which is how many spikes have to then be coded. So if you have 100 spikes on the surface of a virus, and and only 10 are required, only 10% are required to function at the same time to enter the cell, then you want to cover more than 90% of the virus with the antibody. But again, this can vary between viruses and between between viruses. So we don't know, but what we can say is that the fraction of antibody that's elicited in response to an infection at the concentration, is typically higher than the concentrations of replicating a virus. So you're usually going to have more antibody present that can bind and attach to the virus than you do actually virus within the person. So with that, right, then you can imagine that most viruses are going to be completely coated with antibodies to help prevent them from infecting cells.
Got it. Thank you. Another question. Can antibodies be designed to avoid forcing SARS-CoV-2 evolution?
I think, all right, so we can engineer antibodies to potentially promote escape through a limited route. And so this is something that, Pamela's group has done in the past before I joined when it came to HIV. And the example in HIV is there's a certain class of antibodies that overlap or mimic the cell surface receptor CD4 that is used for entry for the virus. And because these antibodies bind in a very similar fashion and mimic that receptor, mutations in this site, sometimes can disrupt the interaction that the virus is typically used to enter the cell. And so one idea, of course, is to design antibodies that, drive the evolution towards, escape mutations that are deleterious to the virus, right? That make the virus inept at entering cells. And basically less fit. So this is a great idea. I think in practice is somewhat difficult to drive the evolution of a virus, but it's something that people are constantly thinking about and pursuing because yes, that would be the ideal case, especially in something like HIV where it's a chronic infection. And if you could deliver a therapy that would then drive the virus into a certain, less fit state, then that would be, the best way to get rid of it.
Yeah, thanks. Another question, if there are effective antibody cocktails available, why aren't doctors prescribing them? And why do patients need to reach out to these hotlines directly?
So, I can't speak for the doctors, but, I think one thing that can be said is, a lot of them may be just overwhelmed and not really know. So from my own personal experience, with a friend that is a doctor, they were unaware of these treatments. But they're working in the ER, patient facing and seeing COVID patients on a day-to-day basis. And so, in this person's case, they just weren't aware that these treatments were available and that they were, widely available. Because again, at the beginning of the EUA, the emergency use authorization for these antibody treatments, it was believed that they were going to be in short supply. And so many thought, it wasn't actually going to be something that can easily be, found and delivered because of low production. And so I think a lot of people just didn't really, think about using them as treatments. So if you are in a situation where you are at high risk and you are infected and you get a positive, SARS-CoV-2 test, or you have parents who are, older and, have these comorbidities and are at the potential risk of, higher risk of fatality upon infection, then you would definitely, tell your doctor that, you've heard about these treatments. These treatments are effective when given early on. So could we explore ways to get these treatments and infusions? It's you might have to call these hotlines more than once. You're gonna have to reach out to multiple people, most likely. I've heard it's actually been quite difficult to get people on board to prescribe it, but you should definitely push for it and fight for it if you are at risk.
Yeah, thanks for clarifying. So we have time for just one more question, and I think you may have touched on this a little bit during our conversation. The question is, do the vaccines cause the body to generate different antibodies depending on which vaccine is given to a person?
No, well, I can't say with clarity, but I can say from the data we have collected and looked at with the two mRNA-based vaccines, the response of, they are listed and the antibodies that are elicited are functionally similar to that between each other, as well as to, what is seen in natural infection. And that's because, the two vaccines that are currently, being administered here in the United States, as well as the Johnson & Johnson, AstraZeneca vaccines, and Novavax vaccines, they're all presenting the same protein, that spike protein, in a functionally similar way. And so, because they are presenting the same exact protein, then you expect that the response that is elicited by the vaccines are all going to be, near identical. And you're going to induce the same types of antibodies they look functionally the same.
Great. Well, thank you so much, Dr. Barnes, and thanks to everyone for joining us. Be sure to sign up for the next conversation on COVID-19, which is on March 15th, when my colleague Emily Velasco will be interviewing graduate students from the Ismagilov Laboratory on viral transmission. And in the meantime, if you're interested in exploring more of the science behind viruses and COVID-19, please check out the Caltech Science Exchange. The site launched last spring to provide trustworthy answers and clear explanations on critical topics in science and technology. And you can find it at scienceexchange.caltech.edu. Thanks.